Hey there, we're going to go over the notes from our packet, page 173 and 174. So we're going to start with our bacterium. So bacterium have one large circular chromosome that contains its DNA. It has, remember, the DNA is the instructions on how to build proteins and all the material for the cell, or in this case, the bacteria. There is no nucleus in a bacteria. Remember, it is prokaryotic. Bacteria also have what's called an accessory DNA or a plasmid. Plasmids can be shared from one bacteria to another, and because of this, the new DNA in the bacteria can allow the bacteria to create special proteins that it couldn't normally be able to do. And then that's what can cause some bacteria to suddenly become resistant to an antibiotic that it was not previously resistant to. Bacteria also have ribosomes. Remember, we said ribosomes make proteins, and proteins are essential for life. So bacteria and prokaryotes have to have ribosomes. Bacteria also have a cell membrane, which coats kind of the inside. This is kind of the grayish layer. But then they also have a cell wall, which is this highlighter yellow color. The cell wall on the outside is also called the capsule because it's encapsulating all of the bacteria. Is that it? Okay. Let me go back a second just to make sure. Yes, awesome sauce. Okay, next we're going to talk about the nucleus. So prokaryotes do not have a nucleus. We said they do have a nucleoid region where the DNA is found. Now, in eukaryotic cells, the nucleus contains the DNA. And the DNA is like the recipe for life. It's not a blueprint. It's not a layout of a house. It's a recipe. Eggs, milk, butter, flour to make a cake. DNA, A, T, C's, and G's to make you. Inside the nucleolus, which is inside the nucleus, is where we make ribosomes. Surrounding the nucleus is basically a nuclear envelope. You could call it a nuclear membrane as well. Inside the nucleus, we said, is our DNA. When it is folded and tightly packaged around proteins, we say it is chromatin. You're probably more familiar with chromosome, but chromatin is when it is folded and packaged tightly. Last, we have our nuclear pores. These are basically holes in the nuclear envelope or the nuclear membrane that allows for the transfer of molecules such as messenger RNA, which we'll learn about much later. But these pores allow for this RNA to enter and leave the nucleus so that the nucleus can communicate with the rest of the cell and the other organelles. And that's it. If you have any questions, please let me know.